Okay, so we'll start today's class. Good evening, everyone. So we'll start today's live session. My name is Aishi, and this is the second week two live sessions um, of this course, RNA Biology. And in this course, in this live sessions, what we do is basically we uh, check some last year assignment questions and we try to solve those basically, and we try to understand the concept behind those questions so that it's easier for us to solve similar type of other questions as well. Okay, so that's how it is, and we try to keep the sessions as interactive as possible. Oh, sorry, I have a really sore throat issue, so if my voice is bothering, I'm really sorry, but we'll try to finish this class in a good manner. So we'll keep the sessions as interactive as possible. If you want to add add any input, you can just raise your hand or maybe type in the chat box or maybe unmute and let me know, and we can then act accordingly. Okay, so what we'll do is I'll tell the concept of how we move ahead with this class. We'll discuss some questions and we'll discuss the answer to those questions and the concepts behind those questions. Okay, so coming back to the questions, this is the first question for today. RNA world hypothesis was supported by, so these are four options: meteorites, ribozymes, Montmorillonite clay, and Luca. So if anybody wants to answer this question, I'll stop for 1-2 minutes, and if you know the answer or if you want to try, you can just speak out. After 2 minutes, we'll discuss the answer. Okay, so the answer will be ribozymes. Okay, so we'll discuss what is the RNA world hypothesis and what do we mean by LUCA or uh, basically ribozymes. Okay, in the last class we already discussed about ribozymes a little bit. When we talk about the LUCA RNA world hypothesis, it basically suggests that the last universal common ancestor, that is LUCA, the organism from which all life on Earth is descendant, was likely an RNA based life form, meaning its genetic material was primarily composed of RNA molecules rather than DNA used by most modern organisms, implying that life on Earth originated in an RNA world before evolving to use DNA and proteins as primary functional molecules. Right? So, what is the RNA's potential for early life? RNA molecules can act as both genetic information carriers and catalysts like ribozymes, making them a plausible candidate for early life forms. So, what is the evidence from ribosomes? The catalytic side of ribosomes, cellular machinery responsible for protein synthesis, is composed of RNA, further supporting the idea of an RNA based ancestry. Then, how is the evolution to DNA? According to this hypothesis, at some point, DNA with its most stable structure evolved to become the primary genetic material, while proteins took over most catalytic functions. Let's talk about the origin and evolution of life on Earth. So, when did life begin? Quite early in Earth's history. We cannot exactly pinpoint time but can narrow down a time period with three lines of evidence. Stromatolites, which are basically rocks with distinctive layer structure. They look identical to living mats of microbes, layers of microbes and sediment. Top layer uses photosynthesis. Lower layers uses top layers byproducts. Now, these microfossils dating to 3.5 billion years ago, they are difficult to distinguish from mineral structures, and analysis shows that some structures contain organic carbon that is found in at least three sites. Now, when did life begin? Evidence in metamorphic rocks that life existed 3.85 billion years ago. So the low carbon 12 and carbon 13 fraction in rock layers suggests life. And the biological processes, if refer C12 to C13, a low fraction of C13, and non-biological processes have no preference, so find equal amounts. So rocks before 4 billion years of old are rare and hard to find, and time of heavy bombardment ended about 3.8 to 4 billion years ago. So the last devastating impact between 4.2 to 3.9 billion years ago, and evidence suggests that life as long as 3.85 billion years ago existed, and definitely at 3.5 billion years ago. So life rose and dominated the planet between 100 to 500 million years, right? So what are living fossils? DNA can be used as a living fossil. The more alike the DNA sequence between species, the more recent the divergence and extinction of their common ancestors. For example, bacteria and archaea, genetic material not separated from rest of the cell. Eukarya, DNA separated from rest of cell by membrane. Extremophiles that live near deep sea vents or in hot springs, closest to root of tree of life. Now, where did life begin? Land is very unlikely because there's no carbon, no oxygen, no ozone. UV destroys molecular bonds. Now, shallow ponds. So once favored, it's full of organic material. When evaporated, organic chemical concentration increases, making it easier to combine complex molecules, leading to life. Current experiments indicate lack of chemical energy sufficient to support life. Then deep sea vents and hot springs. DNA evidence suggests that early organisms survived in conditions similar to deep sea vents. There's plenty of chemical energy available. Now, how did life begin? Now, simplest organisms today and those dated 3.5 billion years ago are remarkably advanced. So, what are the natural chemical processes that could have led to life? The assumptions are life began under chemical conditions of early Earth. Life did not migrate to Earth. Now, what is organic chemistry on early Earth? In 1920, scientists hypothesized that the chemicals in the early atmosphere, fueled by sunlight, would spontaneously create organic molecules. It's tested by the Miller-Urey experiment, 1950s. Now, the Miller-Urey experiment is the first flask partially filled with water and heated to produce water vapor. Now, water vapor was moved to second flask where methane and ammonia vapor was added. Electric sparks or lightning in the second flask was energy source for chemical reactions. And below the second flask, water vapor or cooled rain and recycled to first flask. This is a C. Now the result is turned brown with amino acids and other complex organic molecules. So this is the entire setup of the Urey-Miller experiment. If you see, this is a cloud formation, primitive atmosphere, there's lightning spark. This is the Earth's primitive oceans boiling. So we have discussed the formation of solar system and the formation of terrestrial planets. Now, what is wrong with the Miller-Urey experiment? So the variations of the Miller-Urey experiment, we'll talk about that. So different mixes of gases to represent the atmosphere, different energy sources like UV, sunlight, in results, all produce amino acids and complex organic molecules. Now, not as much as original experiment, must be more sources of organic material. What are the sources of organic molecules? Chemical reactions in atmosphere. Lab experiments show this is likely. Organic material brought by impacts like chemical analysis of comets and carbonaceous chondrites. They show that they have organic molecules. Then chemical reactions near deep sea vents. Heat from undersea volcano can fuel chemical reactions between water and minerals. So we'll talk about the transition from chemistry to biology. Now, organic molecules are building blocks of life. There's a low probability of forming life even if repeated several times, and intermediate steps of high probability are necessary. Now, what is the search for self-replicating molecule? They walk backward from organisms that live today. So DNA is double-stranded, that's very complicated. RNA is an obvious candidate, but it's more simple than DNA. The hereditary information can serve as template for replication, fewer steps to produce backbone structure. Now, what is this problem? RNA and DNA require enzymes to replicate. So, in 1980s, determined that RNA might catalyze their own replication instead of other enzymes. Early Earth was an RNA world. On early Earth, short strands of RNA-like molecules were produced spontaneously, partially or completely, and RNA-like molecules that could replicate faster with less errors soon dominated population. So the copying errors introduced mutations, ensuring the production of many variations of successful molecules, allowed molecular evolution to continue. RNA world gave way to DNA world. Now DNA is less prone to copying errors, DNA more flexible hereditary material, and RNA kept some of its original functions. So let's talk about assembling complex organic molecules. So organic soup was to dilute to favor the creation of complex organic molecules. Lab experiment with possible solution is when hot sand, clay or rock is placed in dilute organic solution, complex molecules self-assemble. Like organic molecules stick to surface of clay. It increases density and likelihood of reactions, and strands of RNA up to 100 bases have been produced this way. Other inorganic minerals may also have a similar role. Iron pyrite, which is called the fool's gold. There are positive charges on surface which allows organic molecules to adhere. Formation of pyrite releases energy which could be used as fuel for chemical reactions, right? Now, early cell-like structures. So, advantages to enclosing enzymes with RNA molecules. So, its close proximity increases rate of reactions between them and isolate contents from outside worlds. We see self-replication of RNA. Self-replicating RNA acts as a template on which a small protein or chain of amino acids form. Protein acts as a primitive enzyme that aids in RNA replication. So this diagram shows a self-replicating RNA molecule that also serves as a template for making a primitive enzyme that helps its own replication. So if the RNA and the enzymes are isolated from the outside environment inside a pre-cell, then only the molecules in this particular pre-cell will benefit from the new enzyme. Let's talk about early cell-like structures. So lab experiments suggest that membrane structures existed on early Earth. It forms spontaneously, it cools down warm water solution of amino acids, it mixes lipids, fats with water. Now, if you talk about non-living pre-cells have lifelike behavior, so it grows in size until unstable, then split to form a daughter cell. It selectively allows other types of molecules to pass in and out of the membrane. It stores energy in the form of electric voltage. The organic molecules come in left and right-handed forms. Non-biological processes have not preference. So biological processes do have a preference. If both left and right-handed DNA developed, why did one die out? So this is a quick summary. Synthesis of organic precursor molecules, origin of self-replicating RNA, origin of membrane enclosed pre-cells, origin of true cells with RNA genome, and evolution of modern cells with DNA genome. If you have not heard about panspermia, it basically means life originated elsewhere and migrated to Earth. So life began apparently in rock, then kicked off the planet by an impact. So the support is that organic material is everywhere and some bacteria can withstand large amounts of radiation and go dormant under low atmospheric conditions. So there are two schools of thought basically regarding this. So one group of people think that life did not evolve as easily as imagined on early Earth in time scales we have determined. The problem is the entire solar system was under heavy bombardment at the same time. The other possibility is interstellar migration. Rock that is the problem with this is that rock to be ejected out of its own system then fall into ours and hit the tiny planet of Earth. School 2 is that life evolved easily and was everywhere with suitable conditions. Earth was not first planet with suitable conditions. Migration of life from another planet, say Mars, dominated before early life on Earth could. So basically, it means we are Martians. So there are Martian meteorites, both have possible fossil evidence of life on Mars. So now let's talk about the early evolution and rise of O2 or oxygen. So first organisms had simple metabolism. Atmosphere was oxygen free, must have been anaerobic. So probably chemoheterotrophs. They obtain nutrients from organic material, obtain nutrients from inorganic material. Like modern archaea appear to be close to the root of the tree of life. Obtaining energy from chemical reactions involving hydrogen, sulfur, and iron compounds, all abundant on early Earth. Now, natural selection probably resulted in rapid diversification. So, modern DNA has enzymes that reduce the rate of mutations. RNA is not so lucky, more likely to have copying errors. Higher mutation rate in early evolution than now. Now, photosynthesis, most important new metabolic process evolved gradually. Organisms that lived close to ocean surface probably developed means of absorbing sunlight, for example, UV in particular. So, once absorbed, developed method of turning it into energy. So, modern organisms have purple sulfur bacteria and green sulfur bacteria, much like early photosynthetic microbes. They use hydrogen sulfide instead of water for photosynthesis. Now, photosynthesis is basically using water for photosynthesis developed later, perhaps 3.5 billion years ago, first appearing in cyanobacteria like blue-green algae. Byproduct of O2 released into atmosphere changed the world. Now, O2 is highly reactive. All initial O2 would react with rock and minerals in water. O2 could not accumulate in atmosphere until surface rock was saturated. So rocks like 2 to 3 billion years old called banded iron formations to atmosphere had less than 1% of current amount of O2. So rock evidence suggests that O2 amounts in atmosphere began to rise about 2 billion years ago. So clear evidence of O2 near current levels appear only 200 million years ago. So rise of O2 would have created a crisis for life. So O2 reacts with bonds of organic materials. Surviving species avoided effects of O2 because they lived or migrated to underground locations. So many anaerobic microbes found in such locations today. So what do we know about early eukaryotes? Fossil evidence dates back to 2.1 billion years ago. Dates to when O2 was rising in atmosphere. DNA evidence suggests that prokaryotes and eukaryotes separated from common ancestors much earlier. O2 played a key role in eukaryote evolution, and cells can produce energy more efficiently using aerobic metabolism than anaerobic metabolism. So, adaptations of aerobic organisms could develop adaptations that required more energy that would be available for anaerobic organisms. Let's talk about the Cambrian explosions. It's an animal branch of the tree of life. There are different classifications based on body plans. All known body plans made appearance in fossil record in a time span of 40 million years. That's less than 1% of the Earth's age. Okay. And the animal diversity began 545 million years ago. Then obviously colonization of land. Life flourished where liquid water exists. Life on land was more complicated, had to develop means of collecting solar energy above ground and nutrients below. Life in shallow ponds or edges of lakes. Water evaporates. Natural selection favored that which could withstand periods of drought. Now, DNA evidence suggests that planets evolved from an algae. It took only 75 million years for animals to follow plants out of water. This is the graph showing mass extinctions. We have time before present or millions of years in the on the x-axis, and the y-axis contains percent extinction. Now, what are the possible causes of mass extinctions? Impacts. Impact sites found for KT boundary, suspected for Permian extinction 45 million years ago. Active volcanism, climate change, external influence for copying errors, increase in solar particles or radiation hitting surface, local supernova. Primate evolution. Monkeys, apes, lemurs, and humans have common ancestors that lived in trees. So, tree life. Limber arms for swinging between branches, eyes in front of head for depth perception. Offspring would be born more helpless than other animals. So, the emergence of humans. They did not evolve from gorillas or monkeys. It shares a common ancestor that lived just a few million years old ago. So 19% of human genome is identical to the genome of chimpanzee, and 2% difference in genome separates the success of human versus chimps. Also indicates evolution of intelligence is complex. So after hominids diverged from chimps and gorillas, evolution has followed a complex path. Numerous hominid species existed, some during the same time period. All humans are the same species. So the first skull fossils that are identical to modern human skulls dates to 100,000 year old. Our ancestors shared the earth with Neanderthals. It went extinct 35,000 years ago. Now, what are the cultural and technological evolution happening? Have not undergone biological evolution in 40,000 years. Mutation rates are slow. There are dramatic cultural changes, transmission of knowledge between generations, spoken to written word thousands of years, agriculture, etc. There was technical evolution, technological evolution, result of coupling between science and technology. About 100 years between industrial revolution to landing on the moon and generating weapons of mass destruction. So coming back to what we started with, RNA world is actually supported by uh, RNA world hypothesis is supported by ribozymes. So we have already talked about self-replicating RNA. So obviously ribozymes are basically RNA molecules capable of catalytic activity, demonstrating that RNA can function as both genetic material and a biological catalyst. A key aspect of the RNA world theory, that RNA played a central role in early life forms before the evolution of proteins and DNA. So, ribozymes can catalyze various chemical reactions, including RNA splicing, which is crucial for gene expression. So the existence of ribozymes shows that RNA can perform enzymatic functions suggesting that early life forms could have relied on RNA molecules to carry out necessary chemical reactions for cell replication, which we already talked about. The ribosome, a key component of protein synthesis, is also a ribozyme with catalytic site primarily composed of ribosomal RNA. Okay, so in the RNA world, RNA molecules would have served as both the genetic material and the catalyst for chemical reactions. Unlike today, where DNA stores genetic information and proteins act as enzymes. The discovery of ribozymes in the 1980s significantly bolstered the RNA world hypothesis. So, while ribozymes provide strong evidence, scientists continue to explore the complexities of how an RNA world could have functioned and evolved. Okay, so the answer for this question will be ribozymes. Anybody has any doubts regarding this question? I'll stop for 2-3 minutes. If you have any doubts, you can ask me. Then we'll move on to the next question. Okay, so if there are no doubts in this question, we'll move on to the next question for today. The next question is, hydrothermal vents were important to give rise to life forms. Is it true or false? If anybody knows the answer, they can unmute and let me know right in the chat box. We'll wait for 2-3 minutes. Okay, so let's talk about hydrothermal vents and life forms. Okay, so hydrothermal vents are home to many unique life forms, including tube worms, mussels, shrimp, crabs, and fish. So these organisms live in the extreme conditions of the deep ocean near undersea volcano chains, right? So this is a true statement. Okay, so why do you think that this statement is true? So see the formation of they result from seawater penetrating the oceanic crust at these boundaries and being heated by the magma below, right? Socially, water may circulate as deep as below the ocean bottom, and they are found at the boundaries that define the edges of the tectonic plates found throughout the oceans. So they can be formed on sea mounts such as Lohi, because this is also an area where hot magma is coming close to the bottom of the ocean. So vent sites with animals around them are easier to find visually, but basis biases are view of number of different vent types. Okay. So hydrothermal fluid can be as hot as 380 degrees Celsius but does not boil because of high pressure. And cold seawater reacts with hot rocks and is chemically altered. Okay, now the reduction of sulfate in seawater to hydrogen sulfide to invent systems is crucial for existence of animal consume communities at vent sites, right? Sulfur dioxide oxidizing bacteria are the base of the food chain. Now, photosynthesis is basis of energy for all organisms in ocean and on land, except hydrothermal vents. Chemosynthesis is the basis of energy at vent sites. Bacteria get energy from oxidizing hydrogen sulfide. His implications for life on other planets. It will be chemosynthetically based. So hydrothermal vents may be where life originated on Earth, has prolonged implications for searching for life on other planets. Right? So images. So one of the most exciting and significant scientific discoveries in the history of ocean science was in 1977 near the Galapagos Islands of the coast of Ecuador. So at a depth of 2500 meters, ocean explores first sighted hydrothermal vents, openings on the ocean floor from which heated mineral-rich water emerges. The sighting was a phenomenon that had been predicted but never before confirmed. So, to their amazement, the scientists also found that these vents were surrounded by a large number of organisms that had never been before seen. A diverse ecosystem was thriving there in the absence of the sunlight previously thought to be required to sustain all life. So, where do these hydrothermal vents form? So, Earth's tectonic plates move on a hot flowing mantle layer called the asthenosphere, which is several hundred kilometers thick. Heat within the asthenosphere creates convection currents that are thought to cause the tectonic plates to move several centimeters per year relative to each other. So transform full plate boundaries occur where two plates slide horizontally past one another. Areas where tectonic plates are moving apart from one another are called divergent plane boundaries or C4 spreading centers. So examples of these regions include the Mid-Atlantic ridge and the areas in the Indian Ocean. Whereas convergent plate boundaries form where plates or collide with one another. So these are often the location or subduction zones. The ring of fire of the Pacific Ocean, where the Pacific submarine volcanism a plate is colliding with the continental plates. Submarine volcanism at divergent and convergent plate boundaries produces hydrothermal venting. So, how does hydrothermal vents form? So, hydrothermal vents are the result of seawater percolating down through fissures in the ocean crust near spreading centers or subduction zones. So, the cold seawater is heated by hot magma and re-emerges to form the vents. As the seawater is heated, some chemicals such as magnesium and sulfate are removed, while many others such as sulfur, copper, zinc, gold, iron, and helium are transferred to the water from the hot crust material. The water rising out of the vents may reach temperature higher than 400 degrees Celsius, but high pressure in the deep ocean prevents the water from boiling. So, as superheated metal infused hydrothermal fluids rise, they mix with in near freezing seawater. Rapid chemical reactions cause sulfur and other minerals to precipitate. They may form chimneys or towers as well as mineral rich deposits on the seafloor. And so, what lives at hydrothermal vents? When microorganisms, including bacteria and archaea, use the chemical rich soup spewing from hydrothermal vents, the source of energy rather than sunlight, in a process called chemosynthesis. So, these microbes are the basis of the food web that supports life in a diverse vent ecosystem that may include tube worms, shrimp, clams, etc. So, in this environment, animals are adapted to live in total darkness and to withstand extreme ranges of water temperatures, pressure hundreds of times that are sea level, and high concentrations of what would normally be toxic chemicals. So, vents and vent fields can be short lived due to the tectonic volcanic activity typical of plate boundaries, although some areas of hydrothermal activity are thought to have been active for hundreds, so if not thousands of years. Likewise, the biological communities living on and around hydrothermal vent fields can also be long or short lived. Even if a vent ecosystem is destroyed by volcanic activity, new life can quickly colonize or repopulate the area if magma heat source is provided. So scientists' predictions say that life on Earth began approximately 3.8 billion years ago and hypothesize that this first occurred in the ocean's hydrothermal vents. Studying the hydrothermal vents provides clues to help scientists learn more about the origins of the Earth and also particularly other planets. So the answer to this question will be true. Hydrothermal vents are actually important to give rise to life forms. Anybody has any doubt in this question, you can unmute and let me know, or we'll move ahead to the next question after 2-3 minutes. Okay, so there are no more questions, doubts related to this question. We'll move on to the next question for today, which is, which of these do not represent ribozyme? Hepatitis delta virus, ribonuclease P, ribosomes, or RNA loop? I'll wait for 2-3 minutes for you to answer. Then we'll move on with the discussion of this question as well. So the answer for this will be RNA loop. So among these, if we try to see which one of these do not represent ribozyme, among the hepatitis delta virus, ribonuclease P ribozymes and RNA loop, the answer will be RNA loop. So let's talk about the different types of ribozymes. So what is a ribozyme? We know it's an enzyme. That's pretty self-explanatory. We know that it's ribonucleic acid. It's not a protein, right? This thing actually got the 1989 Nobel Prize in Chemistry by Silt Altman and Tom Schechts. Now, as with proteins, we consider primary, secondary, and tertiary levels, right? Primary is the amino acid chain. Secondary is the folding of the amino acid chain. Tertiary level of structure, the three-dimensional level of structure. So the primary acid structure is limited to ribonucleic nucleotides like base, ribose plus phosphate. The common bases are AGCU, that is adenine, guanine, cytosine, and uracil. The uncommon bases are pseudouridine, inosine, etc. Next, secondary structure. So it follows the Watson-Crick base pairing, which forms the helix. RNA usually assumes A-form helices, small pore along helical axis, rungs stack obliquely to axis. Then secondary structure, conserved base pairing interactions result in three stem regions, uridine containing turn and augmenting helix joining stems 2 and 3. Okay. So what is the difference between ribozyme and phenylalanine tRNA? The folding. So if we talk about tertiary structure, catalytic mechanism, this is an intact phosphodiester, and in presence of magnesium metal ions, magnesium coordinated transition, we have a cleaved phosphodiester, right? So new crystal structure shows 5 magnesium sites. So what is the future of ribozymes? In vitro molecular evaluation of RNA plus high throughput screening might lead to ribozyme based therapies. So there are some ribozymes that are actually in clinical trial. For example, the angiozyme, ribozyme designed to inhibit vascular endothelial growth factor VEGF angiogenesis, and we have heptazyme. Ribozyme targets highly conserved sequences of the hepatitis C virus, HCV drug resistance. Then we have HIV gene therapy. The bone marrow sample treats stem cells with retroviral vector, encodes gene for anti-HIV ribozyme, replant treated cells. So in summary, hammerhead ribozyme challenges central dogma. Tertiary structure stabilized by conserved hydrogen bonding regions. Divalent cations are required for catalysis. And ribozymes offer great pharmaceutical promise. Now, what are zymogens? Several enzymes synthesize as larger inactive precursor forms, proenzymes or zymogens. The activation of zymogens involves irreversible hydrolysis of one or more peptide bonds. The biochemical change usually occurs in a lysosome, where a specific part of the precursor enzyme is cleaved in order to activate it. The amino acid chain that is released upon activation is called activation peptide. Now, pancreas secretes zymogens partly to prevent the enzymes from digesting proteins in the cells in which they are synthesized. Fungi also secrete digestive enzymes into environment as zymogens. External environment has a different pH than inside the fungal cell, and this changes zymogen structure into an active enzyme. Digestive enzymes is zymogens. Digestive enzymes trypsin, chymotrypsin, and elastase produced as zymogens in pancreas. They are transported to small intestine as their zymogen forms and activated thereby cleavage of specific peptide bonds. Trypsin is synthesized initially as zymogen trypsinogen. It is cleaved and hence activated in intestine by enzyme enteropeptidase, which is one only produced in the intestine. Once activated, trypsin can cleave and activate further trypsinogen molecules as well as other zymogens, such as chymotrypsinogen and proelastases. The peptidase in the stomach is pepsin, works optimally in the acidic environment of the stomach. So being active at pH 2 to 3, but becoming inactivated when the pH is above 5. The chief cells at the base of the gastric glands secrete zymogen called pepsinogen. Pepsinogen is partially activated by hydrochloric acid, which is secreted by the parietal cells. And this partially active enzymes cleaves the peptide from other pepsinogen molecules to form active pepsin. So this is the entire structure. If you see trypsinogen, trypsin, chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase. So what is the biological significance of zymogens? They're inactive precursor proteins, a clinically important mechanism of controlling enzyme activities. The case of protease enzymes involved predominantly in food dilation and blood clotting. Activation of zymogens by proteolytic cleavage result in irreversible activation. Zymogen forms allow proteins to be transported and stored in inactive forms that can be readily converted to active forms in response to some type of cellular signal. Thus, they represent a mechanism whereby the levels of an enzyme or protein can be rapidly increased post-translationally. Other examples of zymogens include proinsulin, procollagen, and many blood-clotting enzymes. Now, ribozymes. Before the discovery of ribozymes, enzymes which are defined as catalytic proteins were only known biological catalysts. First ribozymes were discovered in 1980s by Thomas R. Schatz, who was studying RNA splicing in the silated protozoan Tetrahymena thermophila, and Sidney Altman, who was working on the bacterial RNase P complex. And these ribozymes were found in intron of an RNA transcript which removed itself from the transcript, as well as in the RNA component of the RNase P complex, which is involved in the maturation of pre-tRNAs. So ribozymes often have divalent metal ions such as magnesium 2 plus S cofactors. So ribozyme from ribonucleic acid enzyme, also called RNA enzyme or catalytic RNA, is an RNA molecule that catalyzes the chemical reaction. So many natural ribozymes catalyze either hydrolysis of one of their own phosphodiester bonds or hydrolysis of bonds in other RNAs. So they have also been found to catalyze amino transferase activity of the ribosome. And investigators studying the origin of life have produced ribozymes in the laboratory that are capable of catalyzing their own synthesis under very specific conditions, such as RNA polymerase ribozyme. And some ribozymes may play an important role as therapeutic agents, as enzymes which tailor defined RNA sequences as biosensors and for applications in functional genomics and gene discovery. So, although most ribozymes are quite rare in the cell, their roles are essential to life. For example, functional part of the ribosome, the molecular machine that translates RNA into proteins, is fundamentally a ribozyme, composed of RNA tertiary structural motifs that are often coordinated to metal ions such as magnesium 2 plus cofactors. There is no requirement for divalent cations in a 5 nucleotide RNA that can catalyze transphenyl alanation of a 4 nucleotide substrate which has 3 base complementary sequence with catalyst. Ribozyme, which means RNA possessing catalytic activity, increases the rate and specificity of phosphorylated bond cleavage, peptide bond synthesis, widespread occurrence in nature from viruses to humans. So in 1989, the Nobel Prize in Chemistry has been awarded to Sidney Altman and Thomas Schetz for the discovery that RNA in living cells is not only a molecule of heredity but also can function as a biocatalyst. So what are the naturally occurring ribozymes? The rRNA, ribonuclease P, cell splicing group 1 and group 2 introns, hairpin ribozyme, HDV, hammerhead. Their catalytic function are also written here correspondingly. For example, ribonuclease P is phosphoryester bond hydrolysis, and the biological function is also noted here. So structural features affect how RNA can function. RNA contains only 4 unique nucleotide bases compared to 20 amino acids found in proteins. High density of negative charges, localization of bases in the interior of duplexes. Nevertheless, the mechanisms of catalysis are diverse and exploit metal ions. Acid-base mechanism, example use nucleobases, small molecule metabolite as a cofactor and substrate etc. tRNA assistance. Usually ribozyme combines several of these strategies. The catalytic strategies appear to be similar. RNA as well as protein enzymes use acid-base groups and metal ions to activate nucleophiles and to stabilize developing charge on the leaving group. So ribozymes also require formation of a specific secondary and tertiary structure of RNA by base pairing of complementary regions. Specific primary structure of certain regions is also necessary. Some ribozymes can speed up the rate of reaction 10 to the power 3, 10 to the power 11 times. HDV ribozyme cleaves the phosphorylated bond as fast as RNAs. For example, if we talk about the metallo-ribozyme, uh, ribonuclease P. RNase P catalyzes site-specific hydrolysis of precursor tRNA, which is essential for the formation of mature tRNA. Catalytic activity depends on the presence of divalent cations, magnesium and Mg2 plus Mn2 plus. Large ribozyme composed of both RNA and proteins. However, RNA moiety alone is the catalyst. So for metalloribozymes, cell splicing introns, large introns greater than 200 nucleotides that are able to splice out themselves in bacteria as well as eukaryotes, pre-RNA protozoan tetrahymena, primary transcripts of the mitochondrial genes of yeast and plants. Splicing intron segments of non-coding RNA that are interspersed among the regions of mRNA that code for protein. Prior to translation, introns must be removed to form a mature mRNA. Self-splicing is unlike common introns. Self-splicing introns can splice themselves out of pre-mRNA without the need for the spliceosome. Although self-splicing introns can remove themselves from RNA in the absence of any protein in vitro, in many cases in vivo, self-splicing proceeds in the presence of certain proteins that increase the efficiency of splicing. Self-splicing introns mediate only one round of RNA processing. So group 1 intron self-splicing is initiated by the nucleophilic attack of 3' OH of an exogenous guanosine on the phosphodiester bond. Group 2 introns nucleophile attack is realized by 2' OH of a specific adenosine within the intron. Now, metal ions are proposed to promote the formation of the correct active site structure, correctly position the substrate, activate the nucleophile by deprotonating the 2' OH of guanosine, stabilizes the negative charge. For example, the group 1 introns, 3' OH of an exogenous G guanosine attacks the phosphodiester bond at the 5' splice site. This bond is being cleft. So G fuses to the 5' end of the intron. First transesterification. The free 3' end of the exon attacks the bond at the 3' splice site. This fuses the two exons and releases the intron. Second transesterification. This is group 1 and group 2 introns. So the G attacks the phosphodiester bond at the 5' splice site. Cleavage between the 3' end of the exon and 5' end of the intron. Terminal 3' OH of exon 1 attacks and cleaves the phosphodiester bond at the 3' splice site. A new bond is formed between the two exons. Intron is released. For this case, the internal A attacks the phosphodiester bond at the 5' splice site. So what is the importance of being folded? Specific primary, secondary and tertiary structure is necessary for recognition for the guanosine binding site. Recognition of the sites of splicing or attack. So group 1 introns as real enzymes. Self-splicing introns mediate only one round of RNA processing. Unlike protein enzymes, but once a group 1 intron has been spliced out, it can act as a real enzyme. It can repeatedly recognize the complementary sequence of another RNA molecule by the internal guide sequence IGS, attack it by 3' OH of the bound G nucleotide and catalyze its cleavage. You can see the summary of whatever we are discussing over here. The group 1 intron after being spliced out and the ribosome attacking the RNA substrate. Now, what are the potential therapeutic use of artificial group 1 introns? We can in vitro change the IGS and thus generate tailor-made ribozymes that cleave, destroy RNA molecules of our choice. So this can be a candidate method for human therapy. So currently synthetic ribozyme that destroys mRNA encoding the receptor of vascular endothelial growth factor is being readied for clinical trials. So it's a major stimulant of angiogenesis and blocking its action may help stave cancers of the blood supply. The small ribozymes of virions and satellites, hammerhead, hairpin, HTV or the hepatitis, delta virus ribozyme. Satellites are small RNA viruses or RNA molecules. Their multiplication depends on the mechanisms of a host cell and on the co-infection of a host cell with the helper virus. Ribozyme is a part of a larger RNA virion or satellite that is being replicated by host RNA polymerases. The product of the replication is being self-cleft by ribozyme activity into unit length RNA molecules. Non-nucleophilic attack of a 2' OH on the neighboring 3' phosphate, forming 2' to 3' cyclic phosphate, probably an acid-base mechanism. 2' OH is activated for nucleophilic attack by abstraction of a proton by a basic group. Another proton is donated by an acid to stabilize the developing negative charge on the leaving group oxygen. In HTV, cytosine acts acid to protonate the leaving group, and a divalent metal ion activates the nucleophile. Saccharide ribozyme. Saccharide and hairpin ribozymes can be found in several satellite RNAs, specific RNA plant viruses, and HDV or hepatitis delta virus, the human pathogen. Co-infection of HDV with HBV is more severe than infection of HBV alone. Now, what are riboswitches? That is the elements of bacterial mRNA that control gene expression by binding of small molecules like coenzymes, amino acids, nucleobases. So, riboswitches may have functioned as metabolite sensors in primitive organisms. So, what are the mechanisms of riboswitch catalyzed reactions? Conformational, like metabolite binding induces a conformational change in RNA that affects transcription, termination, translation, initiation. Okay. So, ribosome is the ribozyme. Peptidal transferase activity can be enhanced by protein L27. However, even in the absence of this protein, reduced activity can still be observed. So, although this protein facilitates peptide bond formation, is not essential for peptidal transferase activity. So, how does RNA catalyze peptide bond formation? Base pairing between the CCA end of the tRNAs. Uh, Sorry, I'll just put my charger. Excuse me. So base pairing between the CCA end of the tRNAs in the P and the A peptidal and amino attack sites at 23S RNA help to position the alpha amino acid for amino acid tRNA to attack the carbonyl group of the growing polypeptide. Sorry. So proton transfer from the amino group of amino acid tRNA via 2' OH of adenosine from the terminal CCA of tRNA in the P site to the O3 accompanied by peptidal transfer to amino acid tRNA. So RNA initially served both as the genetic material and the catalyst. Later, catalytic functions of many RNA molecules were taken over by proteins. This is the RNA world hypothesis that we started today's live session with. Cationic clays such as Mont Morillonite can promote the polymerization of RNA-like polymers into RNA chains. So RNA is the primary substance of Earth, of life. DNA and proteins are later refinements. Okay. So cofactors used by ribozymes include vitamin B12, FMN, glucosamine 6 phosphate. Some of them are used by protein enzymes for oxidation reduction and CC bond formation, and were also RNA molecules capable of something like this, and as some of them persisted up to now. So why do we have protein catalysts? So group 1 interactive site is mechanistical equivalent to DNA and RNA polymerases. What selective pressure led to the current protein-based system for replication and transfer? The reason might be greater fidelity, processivity, reactions rates, functional repertoire, etc. Okay, so we'll not go into much detail. So which of these do not represent ribozyme? That is the RNA loop. So if any one of you have doubts in this question, you can ask me. I'll wait for 2 minutes, then we'll move on to the next question for today. Okay, so if there's no doubt in these questions for this question, then so we can stop the session for conclude the session today here. Okay, and we'll meet again next Monday, same time 7 to 9 p.m. So thank you everyone for joining. Have a good evening. So I will stop sharing the screen. I will upload all this PPT onto the drive, and I'll upload the video recording on YouTube as well. So thank you so much for joining. I'll conclude the session. Thank you.